So you can see here, this is a chest radiograph from a patient with an NKX2-1 mutation. It shows ground glass opacities consistent with lung disease, and a CT scan showing broad-scale hypoattenuation that is suggestive of lung destruction. And also, patients with NKX2-1 mutations show clinical phenotype in the brain and thyroid as well, where NKX2-1 is also expressed. And together, this classical triad of symptoms is known as brain-lung-thyroid syndrome. So now, we're keen to further understand how NKX2-1 operates, with a number of questions going into this dissertation work. So we have some questions including, what are the NKX2-1 dependent gene regulatory networks in early lung development? How does aberrant NKX2-1 activity impact these developmental programs? And ultimately, I hypothesize that in human development, NKX2-1 directly controls core programs for lung program acquisition, and it also suppresses non-lung and disease states. So you can see here in the schematic, NKX2-1 is directly binding to genes associated with these gene regulatory networks that are associated with activation of lung programs and inactivation of non-lung and diseased programs. So it's important to note, however, that mice that are heterozygous for NKX2-1 don't show a lung phenotype. So it's impossible really to study lung heterozygosity with a mouse. So to do this, we really needed to work on getting a human developmental model. As such, we used induced pluripotent stem cells, or iPSCs, as a tool to study early human development. So iPSCs are fully uncommitted stem cells that we can drive to become any cell type. And really, we joke that this is everybody's first baby picture, as we all begin from a small population of fully uncommitted cells. And from here, we're able to expose these cells to the right media conditions, such that we recapitulate all of the major milestones of development. So this includes gastrulation and gut-tube patterning, and ultimately giving rise to an NKX2-1 positive lung progenitor, which is the first lung cell type that emerges in development. And you can see here in our cultures, we begin to see upregulation of NKX2-1. From here, the cells have a very important decision to make, and that is, will they become airway or alveolus? So from here, we can expose our NKX2-1 positive cells to the right media conditions to trigger this proximal distal patterning. One set of conditions takes them to a proximal lung progenitor, which we can further mature into an airway organoid that you can see expresses NKX2-1 as expected, and P63, which is a marker of basal cells. Or we can take them through the alveolar directed differentiation, in which we expose them to a different set of media conditions that will take them to a distal lung progenitor that we can further mature into IAT2s, or alveolar spheres, which are organoids that are comprised of surfactant protein C positive cells. So for the purpose of this talk, just for nomenclature's sake, I'm going to be referring to airway lineages as proximal and alveolar lineages as distal. And we really think that by using this system, we can study human development. So now, we'd really like to study the role of NKX2-1 specifically in lung development. So to do this, the director of the CREM, Daryl Cotton, collaborated with Robin Dieterding, a clinician at Children's Hospital in Colorado, and we were able to generate 3 different iPSC lines from patients with NKX2-1 mutations. You can see them here. One of them is a frame shift in exon 3, one is a frame shift in exon 2, and the final one is a deletion of the entire locus. They're all heterozygous. So all 3 of these patients have brain-lung-thyroid syndrome. They all have early childhood respiratory insufficiency. And it's important to note that all 3 of these mutations are prior to the homeodomain and are expected to disrupt classical transcription factor activity, thereby triggering haploinsufficiency. So really, what we wanted to do is to be able to study the effects of this mutation in a way that controls for genetic background. So to do this, we employed CRISPR gene editing, in which we took a corrected form of exon 3 in addition to a GFP reporter and put it into intron 2 here. This will do 2 things simultaneously. It will correct the mutation, and it will also insert a GFP reporter such that when a cell expresses NKX2-1, it glows green. And we were also able to target the other allele here as well. This gives rise to 2 syngeneic cell lines, one in which the mutation is corrected and the other in which the mutation is retained. This gives us the ability to compare these two lines head-to-head without worrying about other genetic confounders. And we hypothesized by using these cell lines with our directed differentiation platform, we're going to be able to study NKX2-1's role in human development. So first, we performed a very basic set of quality control experiments to make sure that we could turn these cells into lung. So we took these two cell lines and we took them through a directed differentiation process and found that they grow at similar rates. We found that they express NKX-GFP at similar proportions and found that they morphologically look similar as well. So this tells us confidently that we can take these cells and turn them into lung, and also allows us to really compare them head-to-head without worrying about other confounders. So next, we really want to study how NKX2-1 works in health by studying how its mutations make development go awry. So to do this, we really want to take cells at various points in our directed differentiation protocol and probe them extensively. This will generate a sort of roadmap of lung development. Next, we can add on or overlay information acquired from NKX2-1 mutations, thereby allowing us to look at what detours in this roadmap are taken with an NKX2-1 mutation. Therefore, we can use this platform to really study how NKX2-1 works in health. So first, we performed single-cell RNA sequencing, which will identify the entire transcriptome of single cells at a number of different time points, once again. So we have our lung progenitors at day 15, and then proximal or distal progenitors, and then either airway or distal lung organoids. And from here, we're able to present this data. This is called a UMAP. For those of you who haven't seen a UMAP before, all you need to know is that each dot represents a single cell. Two dots that are close together are transcriptomically similar to each other. Two dots that are far away are transcriptomically very different from each other. And when we color the cells based off of their time point, we see the cells cluster very nicely based on time point. This makes sense given that there are so many broad transcriptomic changes that occur across development. When presenting this data by highlighting either proximal or distal lineages, we once again see that they cluster very nicely together. Again, indicative of how many transcriptomic differences there are between proximal and distal lineages. And finally, when we present either mutant or corrected status, we start to see divergence between the two. And taken all together, we can see at day 15, our progenitor sample, that we don't see a lot of differences between mutant and corrected cells. This makes sense because NKX2-1 just had a chance to turn on, and we won't be able to see any effects of the mutation just yet. When looking at proximal progenitors, once again, we see a subtle transcriptomic difference. And finally, when looking at our most mature time point, day 55, we start to see divergence between corrected and mutant cells, though to a subtle degree. Then when looking at our distal progenitors, we already at day 27 start to see strong transcriptomic differences. And then at day 55, we see them fully diverge from each other, really showing us very strong transcriptomic differences between mutant and corrected cells. So this really tells us two things here. It tells us that the effects of these mutations increase across developmental time. It also tells us that impacts of NKX2-1 mutations are much stronger in distal lung development compared to airway development. So next, we just wanted to employ some basic gene expression analysis to make sure that we are making the cell types that we think we're making, basically. And when it's showing expression of NKX2-1, you can see broad expression, and it's important to note that we can identify both the corrected and the mutant transcript. So we weren't expecting to see much of a difference in NKX2-1 between corrected and mutant cells. Then, when looking at some type 2 cell markers, we see surfactant protein C and NAPSA here, expressed specifically in our distal. Also, you'll start to see some differences between mutant and corrected cells that I'll get into in a little bit. Then when showing expression of markers associated with airway lineages, we have basal cell markers here, including P63 and keratin 5, that are nicely upregulated in our airway conditions. And we start to see upregulation of secretory cell markers as well. So this is just telling us that we are making the cell types we think we're making. These were more extensively characterized in the written thesis, so if you want to know more, you can read it. Really, we wanted to kind of dig into what actually these differences are between corrected and mutant cells. So just to show you once again, there's a very strong divergence in the transcriptome at day 55 in these IAT tubes. So for the purpose of this talk, I'm going to highlight specifically the differences that we see at this time point. Of note, there's over 1,200 differentially expressed genes, which is quite impressive. And we see, once again, when isolating these samples, we see broad differences between the two. So now we really wanted to investigate what these differences actually are. So upregulated in our corrected cells, our number one differentially expressed gene is surfactant protein C, which is critical for type 2 cell function and is a known direct target of NKX2-1. When kind of scoring the cells based off of a conglomeration of gene expression, we can see that this type 2 cell module score is nicely upregulated in our corrected cells. Then when looking at what genes are overexpressed in mutant, we see markers associated with lung or with liver fate, including TTR, ApoA2, and ApoA1. Once again, when looking at a module score of hepatocyte-like cells, we see nice upregulation in our mutant cultures. So this is telling us that NKX2-1 is playing a key role in acquiring this type 2 cell fate program, but it's also playing a role in suppressing these non-lung fates. And this has been shown in cancer, so it makes sense. And next, we wanted to take a more unbiased approach at investigating these differences. So we looked at what was upregulated in corrected or mutant, and we put them through gene set enrichment analysis. And we found differences in pathway activation and regulation of beta-catenin signaling, which is critical for Wnt signaling, which is critical for lung development, and also regulation of SMAD2/3 signaling, which are members of the TGF-beta signaling pathway. Then upregulated in mutant, we see transcription factor networks that are associated with FOXA1 and ATF2, which are highly important in liver development. So really one of the most interesting things that have shown thus far to me is that in distal cells, we see these robust transcriptomic differences, and in proximal lung, we see subtle transcriptomic differences. So we wanted to see if we can identify this difference in effect in vivo. So we used a mouse system. So I previously said that mice that are heterozygous for NKX2-1 don't show a lung phenotype. However, we wanted to know if we could tease this apart by employing a more competition-like assay. So to do this, we did blastocyst complementation. So we took NKX2-1 GFP embryonic stem cells. So these are heterozygous. They have one copy of NKX2-1, and the other has been replaced with the GFP reporter, so they glow green. Next, we put that into the developing blastocyst of a normal wild-type mouse with 2 function copies of NKX2-1. So this is just a picture of the microinjection. And really, the cool thing about this system is that you could tell it worked just by looking at the coat color of the mouse. And the thing to note is that these green cells in this schematic come from a brown mouse, and these come from a mouse with a black coat. So you can identify whether or not they are genetic mosaics just by looking at their coat color. So you can see up top, these have an entirely black coat, so they're non-mosaics. And here, you can see a nice mosaic of brown and black, indicating that the experiment actually worked. And this is the surrogate mother, and she's very proud of her work, and I kind of want to make her a co-author. And really, we hypothesized that these NKX2-1 GFP cells that are heterozygous will be outcompeted in alveolar development, but they will be able to compete in airway development. So this is just one representative mouse here. Its coat is 95% brown, indicating a strong contribution by donor cells. This is just a cross-section of the lung. Just to show you the architecture, we can see the airways here and the alveolar space on the outside. So really, based on our hypothesis, we would be able to see more GFP in the airway than in the alveolar space. When looking at NKX2-1 expression, you can see nice airway and alveolar expression of NKX2-1. So next, we observed GFP positivity, again, expecting more green cells in airway than alveolar space. And what we found was a very dramatic outcompetition here. So we could see nice GFP positivity in the airway and nothing of note here in the alveolar space. So this provides useful in vivo validation of some of the some of the conclusions that we drew from our single-cell RNA sequencing data, and shows that for the first time really in lung development of a mouse, that we can tease apart a phenotype in NKX2-1 heterozygous mice. So in conclusion, for part 1 here, I've shown that through single-cell RNA sequencing, we can construct a roadmap of lung development, and we can identify differences that occur with NKX2-1 mutations. We saw a proximal effect that is pretty subtle and a much stronger distal effect. We were able to show this using blastocyst complementation in vivo. And finally, by looking at differentially expressed genes between mutant and corrected cells, we can identify that NKX2-1 plays a key role in acquiring lung or type 2 cell fate, and really plays a strong role in suppressing non-lung programs as well. So really what I've shown you thus far is that with an NKX2-1 mutation, we have an aberrant transcriptome. But we want to get more granular than that and understand how this works. So if you think back to my hypothesis slide, I said that NKX2-1 directly controls expression of type 2 cell or lung fate programs and directly suppresses many of the non-lung programs. So to figure out whether or not these are a direct effect, we compared our differentially expressed genes to previously published data sets that identified NKX2-1's binding sites. And what we found was that actually NKX2-1 targets contribute to less than half of the differentially expressed genes that we see, which is a significant portion, 31%, but it's still pretty surprising to us. So we really wanted to dig into what drives the differential expression of these genes and how do they relate to disease. So what we wanted to do was start thinking about how NKX2-1 could be talking to other transcription factors in crosstalk. And this is computationally predicted binders of NKX2-1. And as you zoom out and see more and more of these, you're very quickly taken into a quagmire of transcription factor crosstalk that's very hard to decipher. So we started thinking about some alternative models. So these include that NKX2-1 could be causing expression of another transcription factor, which could then drive expression of key downstream critical genes. Or NKX2-1 could be binding to other transcription factors. Together they could bind to genomic binding sites, or they could sequester each other from binding to the genome. So to dig in more into this transcription factor crosstalk, we employed a Dorothearegulon, which is a funny name, but really all it's doing is trying to link transcription factor activity based off of expression of target genes. So we were able to use this algorithm, which combines literature-curated resources, ChIP-seq peaks, computationally predicted motifs on promoters, and gene expression inference. So this will, together for each individual cell, be able to score each transcription factor's activity. And when taking the cells through our standard pipeline using transcription factor activity rather than gene expression alone, we still see strong separation between mutant and corrected cells. And we really wanted to dig into what some of these differentially active transcription factors actually are. So you can see this is just the top 50 or so differentially active transcription factors. So we see strong separation between the two. And of note, some of these are really critical for lung development. So with higher activity in mutant, we see SOX9, which is associated with the distal progenitor state. And then we see transcription factors critical for TGF-beta and Wnt signaling, which are two pathways that are really critical for lung development and can be implicated in disease. Next, upregulated in the corrected, we have members of the HIPPO and BMP signaling pathways, both of which have been shown to be very critical in lung development as well. And we really wanted to look for one transcription factor to further investigate that we think is the most interesting. And they're all very interesting, but the one that we ended up choosing is SMAD3 because of its importance in the TGF-beta signaling pathway that's really relevant for disease. So SMAD3 is a critical component of the TGF-beta pathway. So here, you can see the cell here. This is the nucleus, this is the genome. And on the surface, we have a TGF-beta receptor. TGF-beta binds to this receptor and is then able to recruit another receptor, which becomes phosphorylated. This ultimately leads to a phosphorylation cascade that results in phosphorylation of SMADs 2 and 3, which together with helper SMAD4 are able to translocate into the nucleus and drive expression of downstream target genes. However, in the presence of our old friend NKX2-1, Izagaya et al. Showed in 2014 that NKX2-1 actually binds to SMAD3, sequestering it from the genome and preventing expression of downstream target genes. And in the context of lung cancer, if you knock down NKX2-1, the number of SMAD3 binding sites very strongly increases. And with knocking down NKX2-1, it allows SMAD to activate genes associated with the epithelial to mesenchymal transition. So when we saw this paper, our minds were kind of blown, and we wanted to see if we can actually show this in our system as well. So to do this, we did a co-immunoprecipitation assay to identify whether or not SMAD3 and NKX2-1 actually bind to each other. So we used an antibody for NKX2-1, pulled it down, and did a Western blot for SMAD3. And what we found was that in both mutant and corrected conditions, NKX2-1 and SMAD3 do indeed bind to each other. Next, we wanted to be able to integrate this ChIP-seq dataset that identified SMAD3's binding sites with our differentially expressed genes to see which of SMAD3's binding sites are actually differentially expressed. And what we see is that there's a strong number of SMAD3 targets that are differentially expressed, including some genes upregulated and corrected that are associated with type 2 cell function, including SFT83, HOPX, NAPSA, and NKX2-1 itself, which we think is really interesting. And then upregulated in our mutant cells, we see SMAD3 targets that are associated with non-lung endodermal fates, once again, like transferrin, TFF1, and ATF3. So really taken together, we can add another piece of the puzzle here. And you can see here, once again, we have our original bar graph showing targets and non-targets. And now we're able to overlay SMAD3 targets and joint targets, which contribute a significant piece of the puzzle as well. So really taken together, we can see that NKX2-1 directly controls expression of a lot of really critical genes. But also, it can bind to other transcription factors, in this case SMAD3, and prevent expression of off-target programs. So this data is all really interesting to us, but it's important to note that everything that I've shown you thus far comes from one single genetic background. And as such, it's important not to draw these broad biological phenomena from data generated from a single background. So we expanded our studies to include the other genetic backgrounds that I previously explained. So this includes, once again, just to jog the memory, a frame shift in exon 2 here, and also a deletion of the entire locus. And this was the original line that we used. So we wanted to really probe all three of these genetic backgrounds to further distinguish whether or not we're able to replicate this data that we previously found, if we can identify any disease states in these cells, and finally, how modulation of TGF-beta signaling affects these different genetic backgrounds as well. So we took all three of these lines, in addition to a non-diseased control, KOLF2, and we performed my favorite experiment, single-cell RNA sequencing, on all three of the backgrounds compared to the non-diseased control. And really, what we were expecting to see is non-diseased cells to cluster away from all of the disease cells, effectively. However, what we actually saw is that all of the different genetic backgrounds cluster very strongly based off of genetic background alone, which really makes analysis kind of a pain, but is ultimately the data. And really, we wanted to see if we could identify, despite all of these differences in genetic backgrounds, if we could identify a sort of core program that is aberrant here. So first, we wanted to just look at expression of some key genes. So this includes type 2 cell fate, liver fate, hindgut fate, and basal cells. And what we see is pretty jointly that the mutation harboring cells all downregulate these type 2 cell markers, which is expected given that NKX2-1 is mutated in these cells. And then we started to see upregulation of some non-lung endodermal markers. However, we do see heterogeneity between our mutants. So upregulated in BLT1 and 2, we see a more liver-like expression pattern. And in BLT3, which is the full loss, full heterozygosity, we start to see emergence of a hindgut fate. So this is something that we thought was really interesting, and we're not sure if it's because of the mutation itself or if it's because of another genetic confounder, but something that we'd really like to investigate further. Additionally, in BLT3, our heterozygous line, we see upregulation of some basal cell markers, including P63 and keratin 5. This could indicate that potentially for this one particular mutation that there's aberrant proximal distal patterning. So taken together, we do see lots of heterogeneity between the genetic backgrounds, but overall for all three, we see a loss of this type 2 cell function, or fate programs. So next, we wanted to really identify what these core programs that are aberrantly expressed actually are. So what we did was we took each genetic background separately, BLT 1, 2, and 3, and compared them all to our non-disease controls separately. Then we took the lists of differentially expressed genes, and we looked to see what overlap occurs between the three comparisons. So you can see there are lots of differences, once again representing this heterogeneity. But we do see this core program of 219 genes that are consistently differentially expressed compared to non-disease. And when looking through this 219 gene list, we performed gene enrichment analysis, and we found, once again, reassuringly, that we see aberrant bronchiolar and alveolar epithelial cells in lung program, which makes sense given that they are aberrantly expressing a lot of these programs. And also, we saw consistency in that we see TGF-beta regulation is differentially active as well, showing that in all three of these genetic backgrounds with different mutations, that we can see differences in TGF-beta expression. So we sought to do exactly what we did previously, and we saw which of these 219 core genes are indeed SMAD3 targets. So I'm not going to bore you to death by listing all of these off, but one gene that we thought was quite interesting is keratin 8, seen here. And we found keratin 8 to be interesting because it's associated with this transitional state that emerges in fibrosis. So in this schematic here, you can see an alveolar type 2 cell, which in association with some signaling pathways, which includes TGF-beta, adopts this keratin 8 positive transitional cell that acts as an in-between transitional state between alveolar type 2 cells and alveolar type 1 cells. However, in the context of fibrosis or repeated lung injury, this keratin 8 positive state gets stuck. And with repeated exposure to TGF-beta ultimately leads to extracellular matrix deposition and fibrosis. And this has been recently identified in a number of different contexts and diseases, including acute respiratory distress syndrome and pulmonary fibrosis. So we sought to investigate whether or not we could see subsets of our mutation harboring cells that upregulates these transitional state programs. So based off of gene expression that I'll show you soon, we were able to identify different clusters of cells that upregulate this transitional cell state. And of note, we did not see any subclustering of our non-disease controls, and those don't upregulate this state. So then, when comparing our cells to our non-disease controls, we see nice upregulation in all three genetic backgrounds, showing upregulation of this transitional state. And to show this more granularly, this is the top 50 differentially expressed genes from this transitional state in idiopathic pulmonary fibrosis lung tissue. And when showing these top 50 genes on a heat map, we can see our type 2 cell clusters are kind of a dead zone of expression. And in our transitional-like state, we see, especially in BLT 1 and 2, nice robust upregulation of these genes. Of note, BLT 3 here shows upregulation of fewer genes, but still a significant portion. Again, highlighting heterogeneity between genetic backgrounds. So next, we wanted to see if this was TGF-beta driven, and we wanted to see if we could abrogate this transitional-like state with inhibition of TGF-beta. So we treated cells with SV431542, which is a long name, but it's a TGF-beta signaling inhibitor. And so this is the same exact experiment. I'm just overlaying the other data that we generated. And we could see when highlighting all of the samples here, we could see once again they clustered nicely based off of genetic background. However, when we look at each genetic background separately, we could see separation between control and SB treated cells. So this is telling us that there are a broad number of differences between control and SB treated cells. And there are indeed many differences, but for the purpose of time, I'm just going to be highlighting their effect on this transitional cell state. So we can see here, once again, that when looking at our control conditions, that our NKX2-1 mutations upregulate this transitional state. However, when looking at this transitional state in exposure to SB, in our KOLF2 controls, we don't see much of a difference at all. In BLT1, we see a nice downregulation of these programs, and this is also true of BLT2 and 3. So this is really telling us that NKX2-1 mutations cause an upregulation of this disease state, and with a treatment with a TGF-beta inhibitor, that we're able to abrogate this disease state. So in conclusion here, we can see NKX2-1 is very busy. I showed with single-cell RNA sequencing that there are broad transcriptomic differences that occur in alveolar development, and I showed this in vivo as well using a blastocyst complementation assay, showing really directly that NKX2-1 is playing a strong role in the activation of core type 2 cell programs. Additionally, I showed using co-immunoprecipitation assay and further bioinformatics, I was able to show that indirectly NKX2-1 is playing a strong role in suppression of disease states and non-lung endodermal fates. So taken together, this really shows that NKX2-1 is a guardian of the lung fate. So in the future, what I'd like the next generation of bright young scientists to accomplish is really identifying, I was able to identify that NKX2-1 and SMAD3 bind to each other in this system. But what about everything else? There was a deluge of papers in the early 2000s that identified a lot of binding partners that weren't really followed up on. So I think really understanding how NKX2-1 can bind to other transcription factors and modulate their activity would really allow us to further investigate how it works in development. Next, I would really like to see how NKX2-1 could potentially modulate chromatin and affect epigenomics. And finally, really all of this work together will allow us to further investigate how NKX2-1 can operate as a sort of signal tuner and is able to suppress disease states. So before we move on to questions, I would like to acknowledge everybody who's helped me along the way. So really, so much of this is so collaborative, and I have way too many people to thank, but first I'd like to thank George, Daryl, and Gustavo for founding the CRIM, because they really created an environment where collaboration isn't just encouraged, but you gotta do it, it's great. So thank you so much, guys. And really of note, this blastocyst complementation data was generated from a really effective, almost assembly line of myself, John Mukandawire at the Mouse Embryology Core, Michael, Anat, and Jake. So really, we were a highly functioning group and we were able to generate some really cool data. Also, really of note, I'd like to thank Mary Lou, who really, for a number of years, was our lab manager, and she was the NKX2-1 of the lab. She kind of really did everything and taught me really everything that I know. Next, I would like to thank Gene for gene editing these lines, enormously helpful. I'd like to thank Laertes especially for being really an being an encyclopedia of lung development, and for really being able to identify, was the first to identify this finding that NKX2-1 heterozygous cells are outcompeted with blastocyst complementation, so thank you for the tip, Laertes. I'd also like to thank the computational core, especially Pushpinder, who helped a lot in analyzing a lot of these large data sets and really kept me honest with my own, and I'm very appreciative of that. I'd also like to thank all of the cores that have helped, the flow cytometry core, the sequencing core, the mouse embryology core, for all allowing me to conduct these experiments. And finally, I'd like to really thank the CREM administrative team for really listening to me complain more than anything, probably. I'd like to thank the Dissertation Advisory Committee. Every one of our meetings was super insightful and amazing. I'd also like to thank Angie for filling in last second for being my physically here proxy committee member. And I'd like to thank my funding sources as well. So now it's time for the rest of the acknowledgements. So first, I would really like to thank a source of comfort and a source of inspiration for me over the past 6-7 years, and you've stuck with me through thick and thin. And that is Nantucket Blend Cake. are 3 at the creme. I've drank probably thousands of them over the years. Got me through a lot. But in all seriousness, I'd first like to thank my mom. You've been here since day one, literally. And really, you did all of this alone, and you helped me so much every step of the way. And really, when I was in school and I would come home babbling about DNA or whatever I learned that day, you didn't shush me or yes me to death. You listened to me and you asked follow-up questions. And really, that made me a much better scientist and a much better communicator. So really, Mom, it's all for you. So thanks. I'd like to thank the rest of my family. Every time I come home and visit, it's a whirlwind of chaos, and I wouldn't have it any other way. So I'd like to thank my brother Alex, who's really my partner in crime through a lot of life. I'd like to thank my sister Amanda, who was my first PI in a lot of ways when I was a little kid. We played school. You were very strict with me. And I'd like to thank my sister Sarah as well for being a constant source of inspiration for me. I'd also like to thank Braden, my nephew, who isn't here today, but he just won Student of the Month, so big shouts to him. And every time I come home, you really let me let loose and let me forget about my science for a little bit and focus more on Nerf guns or Fortnite. And then finally, I'd like to thank 2 newer family members, which include my future mother-in-law and father-in-law, Deborah and Gary, who are here today. You've taught me how to enjoy Campari Spritz, and you've definitely taught me more than I will ever need to know about fantasy football. Next, school was great until I met this guy in AP Biology in 2011. And this is us winning Best Science Student at Drake High School. Remains to be our greatest accomplishment, I think. But Rich, really, without you, I don't think I would be a scientist today. You really encouraged me every step of the way and kind of dragged me along with you in a lot of ways. But without you... Next, I would like to thank other friends that I have made throughout my entire life. These include, it's too many people to name, but Alan, Maddie, Kevin, Brian, Dave, Doug, everybody. Really, you're all amazing, and you all helped me forget about my experiments that have failed previously that day. And really, indirectly, I think the most important thing that you all showed me was that to be maximally smart in my studies, I should be maximally foolish outside of my studies. And you've all really helped me accomplish that goal. Next is all of my friends that I've made at BU and at the CREM, whether that's members of the cohort that I survived the first year of classes with, or all of the people that I've met at the CREM. You make coming to work every day really amazing. And I've never woken up and been like, oh, I have to go to the CREM today. It just makes life so much easier and so exciting. And whether we're at work doing experiments together, or drinking Nantucket Blend, or after work getting a scorpion bowl, which I appear to do more than I think I do. You've really made the last few years extremely formative for me. And of note, one person at the GREM has really served as my life mentor over the years, and that is George Murphy, pictured here in various Halloween costumes. George, you've shown me, most importantly, you've shown me that science is for everybody, and that anybody can do science, really. So thank you, George. And you've given me advice about anything, and I mean anything. Also, I'd like to thank everybody else that is or everybody in the Hawkins Lab. So Anat, DJ, Andrew, Jake, Mary Lou, Amelia, you're all amazing. You've all helped me. Nobody has ever told me no when I asked for help. And you listened to me every step of the way. You've given me so much good advice in lab meeting. And again, thank you all so much for this. But most importantly, I'd like to thank our fearless leader, Finn Hawkins. So I'm not the only one getting embarrassed today. Finn, I joined your lab. It was a deliberate decision because of your standard for excellence. And you've held me to this standard of excellence, and you've really helped me reach my full potential. And really, sitting in your office and then planning an experiment that quickly grows completely out of control and is too ambitious. Those were some of my favorite moments of grad school for sure. So thank you, Finn. And I would also like to thank the rest of my committee. Laertis, who moonlights as a DJ NKX2-1 actually. Daryl, Shamita, Ruben, and Angie, who was a last minute addition. And really, before every single one of my committee meetings, I completely worried myself sick because you're all so amazing, accomplished, and brilliant. And every committee meeting, I expected to get torn to shreds and cry. But in actuality, about 10 minutes after I entered the room, I'm always having some of the most fun I've had in grad school, pitching ideas off of you and getting ideas, and you were all so informative for the entire process. And hopefully we get to have some fun over the next 2 hours as well. And finally, I would like to thank my beautiful fiance, Alexa, who we met a little over 5 years ago. And on our first date, I invited her to this very occasion. So I'm glad you're holding up your end of the bargain here. But yeah, really, coming home from lab every day, it doesn't matter how the day went. It could go good, it could go bad, but I'm always feeling loved and feeling worthy, regardless of what's happening in lab. So I'm really excited to marry you, Alexa, and I'm really excited that once all of this is over, I can start pulling my weight for the wedding plan. So with that, we have time for questions, so I'll take any questions. Thank you.
Great talk. I guess near the beginning you mentioned that mouse heterozygotes don't have as much of a phenotype. So I guess after all your analysis, have you speculated all about why NKX heterozygosity is so much worse in humans than it is in mice?
Yeah, that's a really good question. And I think really to be able to answer that question, we would need a stronger head-to-head comparison between, they control directly a lot of the same networks, but really, that's a good question, but being able to determine its binders in mouse versus human, I think would really be able to elucidate that more, yeah. Michael.
I guess you didn't cover, from your single cell of proximal differentiation.
Yeah.
Only saw it briefly, but it looked like there was some discrepancy in the cell type differentiation.
Yes. Yeah, so really some of the differences that we see in the proximal differentiation have to do with more so general proportions of cell types.
Okay.
So NKX2-1's, some of their known downstream target genes include the secretive globins. And really we identified that in the mutations, we see fewer secretory cells that emerge. And that's something we'd like to dig in more with the blastocyst complementation that we haven't got around.
Did you see any of that in the blastocyst complementation?
Yeah, that's on the to-do list here. Yeah, thanks. Stop there.
Spectacular presentation. The patients with the NK2-1 mutations in the lung part of the triad phenotype, is it known if the phenotype affects more the distal compared to the proximal?
Yeah, so that was one thing that we were kind of reassured by in the analysis of the single cell sequencing, because brain-lung thyroid syndrome tends to be a disease of the distal one. However, there are some mutations, so there's differences, and some patients with these mutations have more of an airway phenotype. Of note, one has neuroendocrine cell hyperplasia, and something that hopefully we'll be able to get access to those lines at some point to investigate. Yeah. So 4 o'clock at the Creme, happy hour or sad hour.